Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Tim Anderson. He's a professor and a lead in the program in disease intervention and prevention at Texas Biomedical Research Center in San Antonio. So, Tim, thanks for coming. How you doing?
Very well, thank you.
Yeah. Well, tell me about your work in the program. What's your research about?
So I work on the intersection between sort of genetics, evolutionary biology, and tropical diseases. I specifically work on the two most important of parasites infecting humans. So I work on malaria, vector-borne protozoan, and I work on schistosome, so blood flukes with a snail intermediate host. And I'm interested essentially in using current genetic and genomic approaches to better understand transmission of these parasites, to better understand how drug resistance evolves, how many times it evolves, how we can identify the genes involved in resistance, how we can develop resistance-proof interventions against these parasites. So I hope that gives you an idea.
Yeah. Well, I guess there's a lot of parasites out there. Are you focusing on parasites that we'd encounter in the U.S. or only in Africa? Like, which ones are you focusing on?
So I focus on parasites that currently don't occur in the U.S., but on the other hand, in the last century, malaria was extremely common in the US, particularly in Texas, and was a major cause of mortality. Nowadays, that's not true. So nowadays, malaria and schistosomiasis are distributed in the tropics, but they are still incredibly widespread. They are still an enormous drag on economic growth. So really a major focus for medical research.
So within malaria, it comes from mosquitoes, right? What's the particular mosquito that bites someone and causes them to get it?
So they're anopheline mosquitoes, and we actually do have anopheline mosquitoes in the US that will transmit malaria. So they are still here. There is still the ability for transmission to occur in the States.
So is there any transmission in the States? Is it just a very low level? Like, what is
really, it's at the moment, no. There are occasional outbreaks have been reported from people getting off airplanes or from mosquitoes hitching a ride with a bunch of flowers or something, and then biting people in a florist shop, and you have a small cluster of malaria cases, but then it dies out. So really, there is no endemic malaria, just very occasional flare-ups from importation.
Okay, and then so when a mosquito, gets a blood meal from someone, what does the parasite look like? Is it like a single-celled organism? Is it It's not a worm. It's just like a single-celled bacteria in malaria?
Well, it's not a bacteria. It's a protozoan.
Protozoan.
Single-celled protozoan, eukaryote organism like ourselves. I can take you through. Essentially, in the blood stage, these are parasites of red blood cells. And so they essentially eat hemoglobin. They will divide over a 2-day period and then burst out of these red blood cells, and each parasite will then invade a new red blood cell. So just through essentially eating hemoglobin, you can reach populations of 10 to the 11 parasites in the bloodstream. So enormous numbers and enormous capacity for adaptation to drugs or whatever we throw at them. So these, in the bloodstream, they're asexual parasites, but they produce a few sexually differentiated male and female forms that then can be taken up by a mosquito that feeds. And in the mosquito, these male and female gametes fuse, and they actually, so blood stage parasites are haploid, but these haploid gametes fuse to form a diploid stage in the mosquito. And then you have meiosis and you have recombination. And once again, you regenerate then recombined haploid stages, which migrate to the salivary glands and can be inoculated into another human. And there the cycle starts again.
So by the time someone feels symptoms of malaria and sickness, what's the protozoan load in their blood? Like how many parasites?
Well, again, protozoan load is normally quite high when people feel symptoms. And so symptoms are closely correlated with burdens. But if you have a million or a billion parasites inside you, you are going to start to feel sick. But it's also important to point out that there is an enormous number of people with malaria, say in many African locations, where people have malaria parasites in their blood, but they are asymptomatic. So they are out there playing football, doing all the normal things people do, but they have parasites in their blood and they can transmit malaria. So one of the big questions is trying to understand why people get sick. And why sometimes malaria is asymptomatic, why sometimes it causes horrible symptoms. Again, it's the same sort of question we're now talking about with coronavirus.
Well, so I would guess that someone, gets the malaria parasite in them, and then mosquitoes, don't just magically stop biting them. So I'm sure naive mosquitoes bite them, get the parasite back, and even infected ones bite them, reintroduce more parasite. And has there been any study of that? How many infections a typical person may get with malaria, and does that change the nature of the disease? Like, if I only get bitten by one mosquito and I end up with malaria, will I fare differently from someone else that's been bitten like 10 times and the parasite's been reintroduced multiple times from different mosquito hosts?
Actually, that's a question that my lab is extremely interested in. We can use genetic tools. Essentially, you can take a blood sample from an infected person and you can either sequence or use other genotyping methods, and you can count how many different genetic variants there are at different loci within the parasite. So you can start to get estimates of how many different infections are present within infected humans. And nowadays, actually, with single cell approaches, I have a colleague who has really pioneered single cell approaches to malaria parasite work. You can sequence individual parasites by sorting infected red blood cells into different wells of a plate. And so you can sequence individual genomes and you can directly count the number of variants present. He's found around 20 different variants within individual patients. And we also know, just from sort of epidemiological work, you can count how many infective bites people receive in a single night, essentially by catching all the mosquitoes that land on people, then essentially measuring whether those mosquitoes are infected. And you find that in some locations, in really intense transmission, in places like Tanzania, you can have 40 infective bites per night. Whereas, in other locations, the place I work on, the Thailand-Myanmar border, it's more like one infective bite every year. So you see dramatic differences in rates of infection in different locations. And that actually appears to be really important in driving different patterns of disease that you see. So perhaps
how do people in the Myanmar border that get bitten once a year, how do they fare versus people that are feasted upon all night, 40 and 50 times? Does the disease look any different?
Well, paradoxically, sort of paradoxically, people who get bitten very seldom get extremely ill with malaria. So what seems to be happening is there is no sterilizing immune response against malaria, but there is sort of partial immunity that keeps numbers of parasites down. So in the Thai Myanmar border, where people are sort of have an infected bite once a year, in between times, their immune system forgets about malaria. So each time they get an infected bite, they get very heavy parasite burdens. They get extremely ill, and they need to be treated, or very often they will die. In Africa, on the other hand, when these people are being bombarded by infected bites, their immune system is constantly primed. They can actually maintain low levels of parasitemia that are sort of fairly asymptomatic. So as a result, there's very different patterns of disease. Low transmission, you get high levels of disease. And when you have high transmission, you still do get a lot of disease. So in Africa, it's sort of young kids who tend to die from malaria because they haven't been exposed. They get exposed, they get really sick, and if they're lucky, they survive, and they then but on the other hand, many people die in Africa from malaria. So it's a sort of U-shaped relationship where there's high mortality when there's really high transmission, and there's also high mortality when there's low transmission.
Different factors are acting at both ends of the curve. Yes. So for a typical infected person, how many different variants of malaria will be in them, and how different are the variants amongst themselves? If you sequence them, are you seeing, can you tell, all right, these 4 variants will probably be resistant to these drugs, and these variants are, very different, et cetera?
So, well, actually, so I mentioned my colleague who can now do single cell sequencing. So he is able to actually look at the relatedness between individual parasites within an infection. And you can clearly see that many of those parasites are actually closely related. They are from the same infective mosquito bite. You can see they are essentially equivalent to brothers and sisters. You also see completely unrelated parasites, indicating that they have been inoculated from a different mosquito. So you can actually use patterns of variation to infer how transmission has occurred, whether these variants have been inoculated from one mosquito and they're sort of meiotic products from a single mosquito, or whether they're from different mosquitoes. One of the interesting things are relevant to a question you asked earlier. Even though we see very high levels of transmission in African locations, and we certainly see multiple different parasites within people, very often we see related parasites within people in Africa, suggesting that really that infection is dominated by parasites inoculated from a single mosquito.
Well, if they reproduce asexually, that would mean that if I'm infected by malaria and I have, 10 different variations of it, that essentially those probably would represent competing populations to infect me and take hold, right?
Can you rephrase that?
Yeah, so, let's say, again, I have 2 different variations of malaria inside of me. I've been bitten by a mosquito, and they're asexually reproducing. So they're not mixing. The 2 variants are not mixing and combining and making a third variation. They're staying these 2 separate lines. So when they're within me, is there a competition like variant A wants to dominate over variant B. And so they're reproducing like crazy and trying to beat out variant B. And variant B is doing the same thing with A. Just like you'd have like a heterogeneous tumor, it seems like you have a heterogeneous infection of multiple different kinds, again, that may be competing, and the asexual reproduction keeps them separate.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
That's absolutely true. So when you can actually measure how genotypes are competing, essentially by just measuring frequencies of those different genotypes over time. And we can do that, you can do that in patients, or you can do that in the lab. You can grow parasites in the lab and measure competition between different genotypes. And particularly, we know there are factors that strongly influence competitive ability. Drug resistance genes tend to carry a big fitness cost. So when there is no drug treatment, those resistant parasites actually lose out to wild type parasites. And we can actually see that in the field when chloroquine treatment was stopped in Mali or in Malawi. You could see that the frequencies of resistance genes dropped rapidly over a 10-year period until wild-type parasites, again, were 100% of the population. So that's just showing strong fitness cost of these resistance alleles.
So treating someone with a malaria drug may not directly kill the parasite, but it may cause them to change genetically so they're less able to infect you, the host. If I take a malaria drug like chloroquine, the chloroquine, are you saying it may not work by killing the parasite inside me? It may work instead by altering the parasite so that it's fighting off the chloroquine, and now it can't do as good of a job as infecting me because it's busy with that. And so it's less able to do its job and proliferate, infect me, and kill me.
So certainly when you treat a patient with chloroquine, then those parasites that carry mutations conferring resistance will spread, and they will outcompete the wild-type parasites. However, when you remove chloroquine pressure, those resistant parasites are at a big disadvantage. And certainly in Africa, we see that they disappear over time simply because they are not competitive without chloroquine pressure.
So in analyzing how these drugs affect malaria, you can't just say it's anti-malarial. You have to correlate it with the specific strains to see which strains are most amenable to the drug's action, right?
That's exactly the case. So, one of the big reasons for trying to understand drug resistance is it means we can genotype parasites from people's blood. We can understand whether a particular drug will work in a particular location simply from the frequency of infections containing resistance genes. And that's using those kind of molecular approaches is far quicker than doing big clinical trials to see whether people get cured or not. So essentially, by genotyping parasites, we can get a pretty good profile on which drugs they are resistant to. And certainly nowadays, in the place where I work in Southeast Asia, parasites have resistance to 5 major compounds that we've used to treat them over the years.
So is there a profiling? Is there a list of the variants that have been observed in the past and which drugs they're susceptible to? I mean, now malaria treatments probably should be a cocktail. I would think that, when someone's sick, you'd try to sequence their parasites, look at the load of each, and then prepare a cocktail that's specifically, concocted to get rid of those variants and their predominance.
That's pretty well what we do. It's slightly less personalized than that. We can really just identify different locations of the world, and we can profile the parasite populations in those regions. And so we can say in Southeast Asia, we'll use, or on the Thai-Myanmar border, we'll use a particular combination. In Cambodia, we might use a slightly different combination. And in Africa, we might use a different combination again. So you can certainly, you can identify combinations that work in different locations. But it's becoming increasingly difficult.
Well, how does this resistance arise? What's the mechanism for that?
Mechanism, like all resistance, is mutation and selection. So I mentioned earlier on, we can have 10 to the 11 parasites in a single infected person. That's a lot of parasites, and every time these are replicating, there will be, say, 1 in 10 to the 9 or 10 to the 10 bases will be incorrectly copied. And so when you sum that up, within every single infected person, you will have 100 different mutations at every position in the genome being generated. So really, there's all the raw material for then selection getting to work and selecting mutations resulting in resistance.
But can you watch drug resistance happen during an infection, or it's just the surviving variants that have the resistance?
It's actually,
or does it develop?
Yeah, you can certainly, you can't watch it so much happening during an infection, but over time, you can watch resistance alleles spreading through a population. So, for example, we have documented a complete selective event from the origin of resistance mutations to spread of artemisinin resistance to around 95% frequency on the Thai-Myanmar border. And that happened over a period of just 15 years. So things can happen really quickly. It's not quite like HIV or other viral infections where you can really document within a single infection resistance alleles arising and spreading through an infection. And in fact, one of the things my lab has been really interested in is trying to understand how many times resistance actually evolves. In that, if resistance evolves very commonly, then you need quite complicated drug cocktails to prevent it from being able to arise. But if it arises really rarely, then the problem may be slightly easier to deal with. And I mentioned to you before that, we have enormous numbers of parasites, enormous numbers of mutations that potentially can arise. However, when we look at resistance to pyrimethamine or to chloroquine, we actually see there's remarkably few independent origins of resistant alleles. So high-level resistance to pyrimethamine, which is a drug that attacks the folate synthesis pathway, that occurred just once and spread right across Southeast Asia. And then that resistance mutation hopped over into sub-Saharan Africa and spread across sub-Saharan Africa. So, we see sort of quite few origins of resistance alleles.
So, again, what does resistance look like? If a drug, all right, first of all, what are the mechanisms by which the drugs stop malaria? Do they, cause the protozoan membrane to open up and to burst? I mean, what? And then when they become resistant, what happens? Are they making like efflux pumps like bacteria do? What does this look like?
So it happens in several different ways. So I mentioned the drug pyrimethamine. So that is a competitive inhibitor of an enzyme in the folate synthesis pathway. These parasites need folate to grow. So you treat with this drug and they are no longer able to make folate, they can't grow. So the mutations that occur in this in this enzyme, and there's 4 different mutations that occur with increasing levels of resistance. They once again they prevent the drug from effectively binding, and so the parasite once again can make folate and can survive drug treatment. So this is a case where you're interfering with the ability of parasites to essentially make nutrients, and they have a workaround simply by changing the enzyme. In the case of chloroquine, when parasites eat hemoglobin and they digest that, and when they digest it, they make toxic side byproducts, heme, and that heme is polymerized into a non-toxic form by these parasites. So chloroquine essentially prevents polymerization of these waste products. So parasites die in their waste products. And so there's another example of interfering with with nutrition. In the case of artemisinin, parasites, again, it's to do with hemoglobin digestion. The drug artemisinin, which is currently the major drug used for malaria, it kills parasite. Well, the drug is activated by the byproducts of hemoglobin digestion, and then it kills parasites using free radicals. And so what parasites do, resistance involves a gene that's involved in uptake of hemoglobin. And so mutations in this gene prevent hemoglobin from being taken up in large amounts. The drug is not activated. It then doesn't kill those parasites.
When malaria enters into a red blood cell, does the red blood cell still function normally at all? Does it take up oxygen? Does it, then provide it to cells, or is it just kind of a zombie that goes throughout the circulatory system but now doesn't do its job anymore?
Essentially the parasite enters red blood cells and it completely remodels the red blood cell to suit itself. So those parasites really become just mobile stores of hemoglobin for the parasite to eat through. They no longer, I don't actually know whether they can still carry oxygen. I imagine when the hemoglobin is gone, they cannot. So.
Yeah, I just wonder if they still participate in their normal jobs, or again, they doesn't happen. What about in the spleen? Have people looked at people's spleens when they had malaria? And I guess the spleen is the eater of no longer working blood cells. So maybe there's a lot of activity there.
I think there's a great deal of activity there. That's not actually my field. I know that when parasites actually or infected red blood cells go through the spleen will actually pick out parasites from those infected red blood cells and kind of reseal those blood cells.
It's amazing that it does that, but interesting.
Yes.
Okay. So where are you trying to make headway here, in terms of drug resistance? Is it really on the understanding of it, or is it the creation of new drugs? I mean, what's your end goal?
So I think several goals. We'd like to, as I mentioned, better understand evolution of drug resistance. So we're interested as well in the idea we talked about earlier about fitness costs of drug resistance genes, and how parasites get around those fitness costs. So I mentioned that parasites in Southeast Asia may have resistance to multiple different antimalarials, and each one of those resistance alleles will carry a cost in terms of fitness. So you wonder, how do they actually manage to survive at all? There's a lot of work in the bacterial field showing that when resistance alleles arise, there is compensatory evolution elsewhere in the genome that will help to restore fitness to those resistant bacteria. So one of the things my lab is extremely interested in is trying to understand whether compensatory mutations also occur in malaria parasites. And yeah, we have several ways to look at that now. Nice thing about malaria is we can actually now keep the whole life cycle in the laboratory. So this is a human parasite, and the only way to actually use the human part of the life cycle previously was either to grow parasites in flasks or use chimpanzees. Chimpanzees are no longer allowed, but we can now make humanized mice with human livers. So we can infect mosquitoes, we can have those mosquitoes inoculate these mice, we can have the parasites go through the liver stage of the life cycle and enter the bloodstream, and we can then take the blood from that mouse. We can put those parasites into culture, and yeah, so we can really keep the whole thing going. That means we can actually now do genetic crosses in the laboratory, and we have excellent collaborators in Seattle that conduct these crosses. So we can actually now pinpoint genes involved in drug resistance or involved in compensation, in growth of parasites. So we can really get a better idea of the different genes that are involved in developing a drug-resistant parasite. So we're very excited about that.
Do these parasites, these protozoa, can they experience epigenetic changes? Are they able to alter their gene expression?
They certainly can, and there's good examples of epigenetic changes in expression of variant surface antigens on the surface of Plasmodium. So certainly epigenetics is important. In terms of drug resistance, it appears that most of the changes we see are hardwired genetic changes involved with sequence changes.
Are epigenetic changes responsible for any drug resistance, or is it just it's at the gene level only?
Well, I think it's early days. I think some people would say yes. I'm inclined to say that the vast majority of drug resistance is hardwired genetic changes in DNA.
Okay. Well, very good. Tim, what's the best way for people to find out more about your research and keep tabs?
I can certainly provide a website, and I suggest searching on PubMed for Tim Anderson, Malaria, and Texas Biomed, and that will give them what they need.
Okay, excellent. Well, Tim, thanks for coming on the podcast. I appreciate it.
Okay, you're very welcome. So we didn't actually get on to the other parasite that I work on. That's a pity.
Well, that just means that if you're up for it, I'll have you back and we can talk about the next one if you're okay with it.
Sure, I'm happy with that.
Okay.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.